I think people remembered last year there was a lot of excitement around Ebola. It was the new hot new virus, and who knew that we'd have yet another new virus this year. I don't think we'll get this every year that we'll have the virus of the year. And in fact, I think this virus may have a little more staying power given the mosquitoes, and we'll hear a little more about that. So I don't think this is the last time we'll hear about Zika virus. But we have a great panel today, and I'd like to thank all the panelists for coming to visit. Marion needs no introduction since she just gave a talk to us here. But the other panelists, I'd like to have them introduce themselves. We have Lark, Coffey from UC Davis, and Daphne Ma from Gilead, and Eva Harris from UC Berkeley. They're just going to talk for a minute or two about what they do, their particular field of expertise, and how it relates to Zika virus. And then we're going to open it up to you, the audience, to ask questions, and I'm sure there are going to be a lot. So without further ado, Lark, can you start with what you do?
So I'm Lark Coffey. I'm an assistant professor in the School of Veterinary Medicine at UC Davis, and my background is studying the ecology and evolution of mosquito-borne viruses, and I'm particularly interested in the viral genetic changes that mediate emergence through increased transmissibility by mosquitoes or increased pathogenicity in people. In terms of Zika, we have developing research projects. So I co-direct surveillance of California mosquitoes for the arboviruses that are in the state. So naturally, we're very curious to and developing competence to be able to detect Zika in California mosquitoes. In concert with that, we're also performing vector competence studies in the laboratory to assess the potential for local and North American mosquitoes to transmit Zika virus. I'm also working with collaborators at the California National Primate Research Center to develop a non-human model of Zika infection, namely in rhesus macaques. So we've been performing experimental infection of macaques with a 2015 isolate of Zika virus and then characterizing infection outcomes. So we've done that in non-pregnant animals, and we have ongoing pregnant animals infected right now. So I'm happy to talk about those studies if you all have questions.
Hi, I'm Daphne Ma. I'm a research scientist at Gilead Scientists, which many of you may know is just down the road in Foster City. So I am the biology lead for a new initiative that was very recently created and supported by our upper management at Gilead Sciences to look at antiviral therapeutic development against neglected and emerging diseases. And my focus is mainly on insect-borne viruses and pathogens. And my previous background was mainly in academia. I looked mostly at West Nile virus pathogenesis and immune responses. And so I'll be happy to talk about our initiatives that we are starting up here at Gilead and how we view the development of drug therapeutics against Zika and other flaviviruses.
Great. So I'm Eva Harris here at the School of Public Health and Division of Infectious Diseases and Vaccinology. So we have, for the last 20 years or so, we've been working on dengue and then chick and now Zika. But my program is pretty broad, and it goes from virology to immunology, pathogenesis, and animal models, and then clinical diagnostics and epi studies and control. So the last piece is through about 30 years of on-site collaboration in Nicaragua in Central America. So that whole program just went into Zika. So we are doing actually a lot of stuff right now. I was just going to mention something related to a question that came up about the enhancement. We have a mouse model for dengue and decided not to get into the fray to the 10 labs that immediately made the same mouse model at once. but we ended up being asked to do figures for someone's paper where we just now showed lethal enhancement of dengue infection by anti-Zika human monoclonal antibodies. So going the other way, that is possible. It doesn't mean that it will always happen, but it is possible, and therefore there's issues about vaccine development. It's just something to keep in mind. Another thing that in terms of the pathogenesis work that we do, we have some really interesting new findings about a non-structural protein 1, NS1, that's secreted by dengue infected cells, and we've been looking at that NS1 protein from a lot of different flaviviruses, so dengue, West Nile, and yellow fever, and Japanese encephalitis. We found some really interesting tropisms for causing permeability, which is essentially vascular leak on endothelia from different cell sources, and we've actually looked at Zika NS1 and found that, whereas dengue dengue, for instance, causes endothelial permeability in human pulmonary cells and a number of other types. Zika does not cause permeability in pulmonary cells, but it does in umbilical vein cells, as well as in brain microvascular endothelial cells. So that's kind of something interesting that we're starting to follow up. More broadly, we've involved very much in different diagnostic methods. And so we have a very long-term cohort study, 12-year cohort study in Nicaragua and a 17-year hospital-based study. And so we've added Zika to that. And so that's allowing us to look at a number of really important aspects, one of which is to develop new diagnostics, which we have a triplex Zika chick dengue assay, and have learned a lot about viremia and like the kinetics in urine and stuff like this that we need to know. But the other is that it's a huge mess on serological diagnosis because Zika and dengue cross-react, and it's essentially, excuse my language, but it's screwed the entire Latin American surveillance systems for time immemorial, and of course our studies as well. So we're desperately trying to find next-gen serological methods that we're not developing because that's not where, essentially we already have a number of colleagues who have, peptide arrays with Ian Lipkin and, with Fremont, and they've got new, like, knockout fusion loop envelope proteins. And I think that the only way forward is going to be to try and find new kind of clever ways to get around this cross-reactivity. So that's kind of a big aspect. Our cohorts also lend themselves really importantly to the question of whether there's a clinical or immunological enhancement of dengue to Zika. In other words, and not only, because we have 12 years of immune history of these children, so we know kids that have had no dengue, one dengue infection, or two or more dengue infections. And it could be that the enhancement possibility is different when you've had one or two or more dengue infections, because if it works like dengue does, then that's going to be something we can find out. And so we've essentially gotten, NIH has kind of asked us to add Zika to a number of our ongoing projects. So we have a big systems immunology, human immunology project consortium where we just have added, first it was dengue, then chick, and then Zika, which allows us to do CyTOP and Luminex and RNA-seq and these network models. And interestingly, we can now compare them, not just Zika plus and minus dengue, but we can also compare it to dengue and chick and Zika. So, all of this is just kind of, and we've gotten involved very much with a big, huge pregnancy study that's multi-centered for a number of Latin American countries, and NIH is working on, so that's been a huge amount of work. And then finally, we do a lot of work in communities, looking at evidence-based community-derived interventions for mosquito control. And really, we did a big cluster randomized control trial on like 80,000 people in Nicaragua and Mexico, and showed that you can, just by people understanding the life cycle of the mosquito, can actually get rid of where the mosquito breeds and be able to reduce not just the rates of mosquito infestation, but also infection measured by children's antibodies in their saliva to dengue. And so then there's another offshoot of that that's called dengue chat, which is now chick chat, which is now Zika chat, which is essentially a cell phone application, and brings people in, especially adolescents, into not just going in their cell phone, but actually, getting rid of where the mosquito breeds and then documenting that, and that's how you become a dengue warrior, or a chick warrior, or now a Zika warrior. And I feel like that's really important. I'm talking to people on the boards here, because it's so important to get people involved, because it's a mosquito, right? I mean, Zika is a problem as a virus. It's a big fat problem, but it's all being transmitted by the same mosquito. So we have to both do the fancy virology, but then we just got to get rid of where these mosquitoes breed. And so any way we can do that, I think is really important. So any questions along that line, I'm happy to answer.
Okay, A to Z. Okay, so do we have any questions from the crowd here? Yes, Peter, you can.
Yeah, I'm dying to know. So, if you're infected with Zika virus, are you immune to reinfection?
To reinfection. That's something that. Does anyone else want to answer? I mean, we don't know yet. We're assuming that it acts kind of like dengue, like dengue reinfection. We actually have just shown in a new paper that we can get homotypic dengue infections, meaning with the same serotype twice, with a log of up to 6-fold viremia, but it doesn't really change dramatically the clinical outcome at a population level, but you can get reinfection. And I don't think with Zika you would get breakthrough clinical infections, and it's essentially one serotype. Even the Asian and the this and that are kind of one serotype, as opposed to, they're different genotypes, but they act immunologically as one serotype. So I don't expect that we'll get reinfection, but that's precisely what we want to do over the long run, is continue our cohorts and look for that. And that's the only way I think we'll be able to answer that, although I think you can also answer that really well.
So I think most of your questions today, we are going to say, we don't know. That's the truth, and that's sad, but we can guess, and we can guess from our past experience with other flavivirus, including dengue. So I think this cross-reactivity with dengue is an issue in terms of vaccine, obviously, and also in terms of diagnostic, and the question of persistent immunity. So without consistent follow-up studies where you have access to samples that are collected longitudinally post-infection and over years post-infection, you wouldn't know for how long the IgM titer persists, if they develop, and if the IgG response persists, and neutralizing antibody persistence and protection. So I think for now, we don't know.
Okay, next question.
So I would like some help from the panel. I'm more interested in real-world effects. I'm not a virologist. I'm a nutrition biochemist. What I noticed was that a child born with a glucose transporter 1 deficiency, in other words, the glucose can't get across the blood-brain barrier into the developing fetus, they develop microcephaly. So I tried to see, what about the pathogenesis? Maybe we're dealing with a viral effect on allowing energy to get into the fetal brain. And glucose, GLUT1, which is what it's called, is also the main way that energy gets across the placenta. It's also the main way that energy gets across the blood-brain barrier. Perhaps we may be dealing with an acute neuroenergetic deficiency. And this is why the child's brain doesn't develop normally. It doesn't get the energy it needs. And one of the things Dr. Lanteri talked about the previous outbreaks of Zika in French Polynesia, Yap Island, and so forth. We did not see the same level of microcephaly that we're seeing in Brazil. For me as a nutritionist, that brought up the idea, well, coconut is an important part of their diet. Coconut has medium chain triglycerides, which are metabolized by the liver into ketones, and ketones can get into the brain without going through that GLUT1 transporter. So are we dealing with an energetic problem here? And perhaps there might be a way to provide this alternative source of energy to women who are pregnant and maybe give their body's immune system enough time to get the upper hand.
So first, let me correct something here. I think you mentioned that during the outbreak in French Polynesia, we didn't see this effect on microcephaly as much, right? But here we are dealing with a problem of number, right? French Polynesia as a territory is very spread, it's islands, but the population is limited compared to the population of Brazil. So we know that this Zika outbreak, Zika can spread very fast, and I mean, there's a ratio of population density and mosquito density that made it really explosive in Brazil. But I think we cannot really say that there was no increase at such level of neurologic disorders in French Polynesia. They did report an increase of this Guillain Barré syndrome and of microcephaly as well. Microcephaly is a very rare event, and they saw it. They reported it, and so I think now it's... Yeah, whatever.
And just one other factor, dengue affects GLUT1. Dengue also affects protein kinase C, which is integral to GLUT1 functionality. So we have like a mechanistic background. This isn't just rolling dice here.
Right. So I just clarified this little thing you started with, right? So now I'm going to give my point of view on your research and your comment. I think this is an interesting comment. I think as we don't know anything, we need to be open to everything. I think there's no treatment to mitigate the effect of Zika during pregnancy. So if you tell us that it's good to eat coconut, I would say eat coconut, right? And if we can take ketones or whatever to compensate, to try to prevent that, yeah, why not?
I was going to just say that I just recently went to Brazil about 2 years ago, and there was plenty of coconuts actually I found on the beach. So, in my perspective, I mean, I was just in Rio, but they have plenty of coconuts in their diet. So,
it's a staple. Just one final thing: the paper came out in the Lancet. You can read it there, and they don't eat as much much in the area of Brazil where we saw the most microcephaly do not eat coconuts, and it's a staple in Polynesia.
Guys, one other thing. I think what's really important here is that what has been established is that there is a causal link between Zika and microcephaly. What we have no idea about is the effect modifiers and the extent of risk. We don't know the extent of risk in a symptomatic infection, and we know even less about an extent of risk in an inapparent infection or an asymptomatic infection, and that's exactly why, these pregnancy studies, the one that we've been involved in is just enrolling, 10, 12,000 pregnant women in, multiple countries and just having exactly the same harmonized protocol to go forward so that we can control for all kinds of things. And I think there's for sure some kind of genetic backdrop that's associated. There could be pesticides that are associated. It could be a numbers game. It could be the dengue, pre-infections. I think there's a lot of different scenarios, and I bet you all of them are functioning because that's essentially the way complex infectious diseases work. But what's really important is that it's not just the NIH study, but there's also at the WHO level, there's actually a meeting in Mexico in 2 weeks where it's all of the different consortia are coming together, the EU, the CDC, the PAHO, et cetera, and NIH, to try and harmonize all of the consortia as well. So there's been this huge worldwide effort in a really beautiful manner to kind of work together so that we can exactly try and understand, the same protocol across all of these countries, where we can just be able to exchange results across countries to get exactly at those kinds of questions. And so that's we don't have the answers, but at least that's in process.
So going back to this question of spread, perhaps. So how robust is the evidence that only Aedes aegypti is the vector? And, do we want to, Eva wants to talk.
Are you speaking in Central and South America and the Caribbean?
Yeah.
I think not very, because there have not been detailed entomological surveys published. So maybe someone can correct me if they've seen it, but I have not seen a published paper where they describe the minimum infection rates in species that they're collecting in now Zika endemic areas. So we think it's probably Aegypti and albopictus. There have been vector competence studies where mosquitoes in the laboratory are fed with, in this case, the paper published was the French Polynesian strain, which is the genetic ancestor of all the viruses that are in the Americas. And both Aedes aegypti and Aedes albopictus can transmit that strain. The mosquitoes were collected from various different places in northern South America, the Caribbean, and then they also included mosquitoes from Vero Beach, Florida. So when they ingest a high titer blood meal, about 20% of both species from all the different places were capable of transmitting Zika.
So there is a paper from Africa that's it's a class 1 paper that someone projected at a WHO meeting I was at recently, where it shows that there's about 15 field-caught species that are positive for Zika. And then I heard that it was actually 25 species across 7 genera. Our data, Josefina Coloma and my group was just in Ecuador working, and they actually found field-caught culex with positive for Zika, which is super bad news because that's everywhere. That's the West Nile mosquito. That's in your bird baths here. That's, so what we don't know is what's driving the epidemic, but there's definitely a lot of different species that can be infected, and that's why the vector competence studies are actually really important to understand, who's doing what.
Hi. I've heard, and there was a publication in suggesting that in Lancet a couple of months ago suggesting that there was an association with climate change that has exacerbated this epidemic. And I would appreciate some discussion about what might be some of the mechanisms of this, and maybe with some of these other arbovirus infections. Thanks.
Well, up to a certain limit, increasing temperatures can expand the range of the vectors. So potentially move the capable bounds of transmission of Zika farther north. Just a few degree increase in temperature decreases the extrinsic incubation time, which is the period that the virus needs to get from the ingested blood meal that the mosquito takes into her salivary glands to be transmitted. So that's highly dependent on temperature. So shortening the EIP in mosquitoes can augment the intensity of transmission by shortening the length of the cycle between mosquitoes and people.
Sorry, I have a very naive question. So the Zika virus, will it kill the mosquito or not? Or will the babies of the mosquito, they have a smaller brain or not? Sorry.
So in general, arboviruses, when they infect mosquitoes, don't exert a large fitness cost. There are some studies with dengue that show that the lifespan can be marginally reduced. But unlike in vertebrates, arboviruses aren't generally pathogenic for mosquitoes. So once a mosquito becomes infected with Zika, it will probably live the normal lifespan of a non-infected mosquito, and it will be capable of transmitting for the duration of her life.
Okay, I have one, taking the moderator's prerogative here. Daphne, I want to hear a little more about, A, how you go about developing anti-Zika antivirals. So that's the first part of the question. And then B, I think we have a lot of students here, so a lot of them would want to know, how did you go from here to there? How do you make that leap, and what's different about it? So if you could just sidetrack a little bit and talk about that after you talk about how you guys are approaching this.
Sure. I can cite back probably to the way that we generally look and discover antivirals from a commercial or a biotech company perspective is we do have the resources, for example, a compound or reagent library that we can screen for effective antiviral activity. And after a series of preclinical tests, we can eventually take them to the clinic for further development. So I'll bring up the example of the recent success that we have with development and discovery of a compound within our library called GS5734, which some of you might be familiar with as having very efficacious effects on the Ebola strain, I believe in Makona. And so this compound was screened in collaboration with the CDC, NIH, and USAMRIID, who helped us further test whether or not this compound was indeed active against Ebola, and really helped us as a collaborator to really have the ability to look at our libraries and see maybe what compounds may be actually active and effective against these diseases. So we will take kind of a similar approach where it really heavily depends on academic institutions, government agencies, and all of you as researchers to help us also look at the resources that we have and leverage it against whether or not we can fully develop a therapeutic against these viral pathogens. And I will also say that this initiative that I am actually a part of is a rather recent development for us at Gilead. We were able to leverage our existing resources to develop this program that we're really looking at as a non-commercial project. So we're not necessarily looking at it from a money-making kind of initiative. So we are very excited about being able to leverage our existing resources and our expertise in antiviral development to applying it to these neglected diseases. Now I actually very recently joined Gilead, and it's been actually a wonderful experience. I can attest to that Gilead is driven by science and also a commitment at all levels, from upper management to us as scientists, really at treating disease. And for me, I actually don't find too much differences in terms of my day-to-day activities. I'm still in the lab personally. I still get to collaborate with and learn from all you as experts in the academic institutes and government agencies. So for me, it's been a great experience.
Can I just mention about just another approach to the kind of inhibitors kind of thing? So we have a really wonderful collaboration with a group at UCSF. It was actually Lanteri Ferris here in the audience, but we've been looking at Zika infection of primary human placental cells. And so these are Nicaraguan Zika viruses that we've isolated, and then all the work in terms of the placental work is being done in her lab. But what's interesting is the approach was to kind of learn what cells are infected and what are the characteristics of those cells, and then look and find inhibitors that are known because of those characteristics. And we actually have one that prevents like over 99% infection of Zika virus in human primary placental cells, just by kind of having that approach. Now, in terms of like, we haven't gotten into animal models or this or that, but maybe we should talk. Yeah.
Can I go back to the previous question that I didn't fully answer about the vertical transmission in mosquitoes? That's also not the norm in flaviviruses. So most Zika transmission between mosquitoes is horizontal. So a female is not going to infect her eggs, and the fitness cost on the eggs is not going to be reduced if the female is Zika infected.
Yes, two questions. One is with regards to the transmission. You just you said there's no horizontal transmission among mosquitoes. That means so it's not going to go from human to mosquito. Is that correct? And the second question was about what is the state of Zika rapid diagnostics today in terms of the cross-reactivity with dengue and other viruses? And
so in terms of the horizontal mosquito-to-mosquito transmission, are you talking about venereal?
No, I'm just asking about, my understanding of the transmission is the mosquito bites a human and then that goes to another human being and infects the human being. So is there such thing as a mosquito, human to mosquito and then to human? Or how does the transmission actually propagate throughout the environment?
Right, so canonically, a human has to get a high enough level of virus in their blood so that when a feeding mosquito ingests a blood meal, there's enough that it can get through the mosquito midgut into the body, into the salivary glands and the saliva, and then the mosquito has to live long enough to refeed on a naive human. So there have been studies with West Nile that show there can be non-viremic transmission. So imagine if there's, in the case of West Nile, a large reservoir like a horse or a bird that has a lot of mosquitoes feeding on at the time. One mosquito can deposit virus the virus just moves through the blood and another mosquito can pick it up. So the vertebrate doesn't necessarily need to be viremic. But again, that's probably a very small minority of transmission events. The majority are a viremic person to a mosquito back to another viremic person. With the caveat that now for Zika, there's been a description of sexual transmission, both male to female and male to male. So that is largely underestimated in our understanding of the role of mosquitoes in the transmission of Zika, because it's much harder to measure the sexual component.
Except in the US right now.
Except, right, as she said, except in non-endemic places like the US right now, where we can with certainty say that a partner acquired Zika without a travel history from a partner who had been recently in a Zika endemic area. So if Zika comes to the US, it will obviously not be as clear.
Your diagnostics question? Yeah. So the conventional diagnostics, which are IgM and IgG, for instance, they will work in a primary infection, a primary flavivirus infection. But once somebody has been either dengue infected or others, yellow fever vaccine, JEV, all this kind of then it's very, very difficult to have a specific reaction. So you will pick up a flavy infection, but you won't know whether that's dengue or Zika, for instance. In at least 30 to 60% of secondary infections, there's this big cross-reaction problem. And so generally we recourse to what's called the plaque reduction neutralization test, which is supposed to be very specific. But in fact, you also have the same problem with the PRNT. So the CDC is just saying, if you have any PRNT titer to the other one, you can't trust this. And all you can say is it's a flavivirus infection. So it's really problematic because about 80 to 95% of people in Latin America are dengue infected, especially by the time they get to adolescence or adulthood. So it's pretty much like the whole surveillance systems and diagnostic systems don't work. Yeah, so there was a paper published recently looking in Florida, and so they used conventional diagnostics with the assumption that people were essentially primary infections for Zika, and then you can use that. What is very good right now, is the PCR, the real-time or various different PCR reactions. If you can catch it during the viremic phase, then you absolutely have a confirmed positive. The problem with Zika is that, at least in our hands, we see a very abrupt drop. And within about 4 or 5 days, 4 days, it's over. And it's if you're looking at RNA, like genome equivalents, RNA copies, a chick will be from like 10 to 12 logs. Dengue will be 6 to 10 logs, and Zika is much lower. It's like 4 to 6 logs or so. So it's actually much faster and lower. But it's actually more. It's much higher in the urine, starting around day 4 to around day 10. So.
I think lots of companies are trying to develop rapid diagnostics, and that's, to me, one of the positive outcomes of this outbreak is that anybody with any kind of expertise is lending it to Zika.
Just to clarify, so it's not higher in urine, right? It persists longer in urine. It's higher in saliva compared to plasma during the acute phase so those 4 days, it will be higher in saliva, but it doesn't bring anything in terms of persistent diagnostic, right? So once we know for how long it persists in all body fluids, and we found a body fluid that allows us to detect RNA, Zika RNA, for extended period of time beyond this viremic phase in plasma, then we have a better diagnostic, but it's molecular based, it's not antibody based. So there is no systematic longitudinal follow-up right now. So what we know is that we think that in some cases, and you don't know if it's everybody or just selected cases, we know that Zika RNA has been found in semen about 63 days or up to 10 weeks post-disease onset. So when is the disease onset starting compared to the time of transmission, mosquito bite or sexual contact? We don't know, right? So it's extended period of time in semen. It's 10 to 21 days so far in urine. So you can extend up to 3 weeks the time of detection in urine. But I mean, once again, once we have a systemic follow-up studies comparing the persistence in all body fluids, we'll know better, and that will inform diagnostic. Sorry, it's 3 weeks in urine. What's the question? 4 days in plasma.
So I had one question and another comment. How similar are Zika and HCV in proteins? And this is for the person in Gilead. Have you tried any of the HCV compounds to reduce the Zika replication in vitro? That's one. The second is, if we have a cohort of people, mothers who've had children with microcephaly, and if it's true that they can't be infected again, maybe down the road in 2 years, is it possible that they won't have another child who is microcephaly then? I'd just like to hear your comments. Thanks.
Yeah, so we actually, for our project, we're looking at all of the insect-borne viruses in the flavivirus family. So we're looking at our entire compound library and evaluating effectiveness for various pathogens. So this is something that we are very aware of, and Zika is definitely a priority. So we don't have any results yet, but we are currently collaborating with several laboratories to do this. And the second question?
So if I were to favor the direct viral-mediated pathology hypothesis, the mother who has the second child is going to be Zika immune, so her second pregnancy would probably proceed without event. But that's if the virus is directly causing it, and presuming that she can't get reinfected, as Eva was saying. We don't really know, but probably not.
Okay, a question on that side?
Yes, my question is about the Gilliam Burress syndrome. This is described as an immune disease. So could you please elaborate how the Zika virus can produce this? And is there any specific group that is mainly presenting it, or is it known also if the percentage of people presenting this is similar with other viruses, similar viruses?
Okay, so I'm not an expert on Guillain-Barré, but I'm the WHO expert panel on the Zika causal framework, and we just started dealing with Guillain-Barré, so all I can say is kind of parrot what I heard like 2 days ago. But so what's interesting, apparently Guillain-Barré is something which can be caused through autoimmunity. It's by several different viruses and potentially vaccinations like influenza vaccine, potentially. But a lot of very little is really known very well because oftentimes there are sporadic cases. So there's not like a cohort effect that you can study. Campylobacter jejuni is associated with and there was one particular outbreak, I think it was in South Africa, so that's kind of a place that was able to be studied specifically. And then Zika is the other. So where there's, the graphs that I've seen are pretty impressive in terms of the, if you look at, I think Colombia, there's like 5 countries that have reported, you can see the peak of Zika, and then like right under it start coming the peak, like no Guillain-Barré being reported, and then suddenly all these bar graphs underneath, right under the Zika peak. So the question is, what's the delay? And there's this kind of funny, at the individual level, there seems to be a couple days to maybe 1 or 2 weeks of delay, but at the epidemiologic level, there's several weeks. And it might be a reporting artifact, I don't know, but we're trying to figure this out. And so I was listening to the neurologists who were talking about this, and it's interesting because if it's kind of too close, then it actually implies that there might be a para-infectious Guillain-Barré syndrome, not necessarily an immune-mediated one. So then it becomes actually very important to understand the actually temporal relation. I had kind of assumed, like most people, that, okay, well, this must be just the immune version, immune-mediated, and therefore you'd expect, a certain delay. But because actually when you look at the individual cases, it's much shorter than, in fact, other situations, people are still considering a viral, like a parainfectious route, where it's actually virus can be causing the neurological defects as well. So that's kind of unclear in terms of the mechanism, but it's pretty convincing temporally that there is that association. And then there's like 10 different things you need to do for causality, and so there's less information known than there is for the congenital birth defects, but at least the temporal and a couple others are looking like that's enough to have pretty much confidence that there is an association.
Yeah, I have one comment on that as well. So there are a panel of antibodies that typically come up in Guillain-Barré syndrome. And it looks like the one that is associated with Zika virus is the axonal form of Guillain-Barré syndrome. And there are these anti-ganglioside antibodies that are typically found in that. And they've looked in the people that they've studied so far and have not found the typical antibodies that are associated with this form of Guillain-Barré. So again, it's a question of, is there a direct infection of the neurons? Because we know it's a neurotropic virus at this point. Or are there different antibodies that are elicited? And I think it's going to be really interesting to figure that out. So there are research studies going on now to try to suss that out.
As a comment related to the Guillain-Barré, well, there is papers describing Zika virus infection of neuron progenitors that you can go and check, that could be related to the Guillain-Barré syndrome as well. So I have basically 2 questions. So you said the virus is able to persist in semen for months. So my question is, do you know, because you said you detect RNA, but you know how infectious is that virus still after several months?
Persistence, detection of RNA versus infectivity, that's something that needs to be addressed as well as part of these longitudinal follow-up studies. You find, as a diagnostic or screening tools, you can find the RNA persisting for extended period of time compared to the plasma. Now, how infectious is that? I think, I forgot now which ones. I think it's been reported also infectious in semen. So there are different cases. I think it was difficult to show infectivity in urine. It was also detected in milk, breastfeeding milk. It was detected in semen, saliva, and in all of these studies, you have the RNA. Now you need to address the infectivity.
Yeah, the infectivity in the urine could be related to the pH, of course, because flavivirus are pH sensitive. That could cause like non-infectious viral particles found in urine but not in semen because the pH is different. Another question is, based on this all crazy cross-reactivity, dengue, flavivirus against Zika, so how can you tell us about like a vaccine, Zika vaccine development? How it's gonna be? It's gonna like, no words, bad words from Dr. Eva Harris, group up everything more, or what is gonna happen?
It's kind of a mess. I mean, I got this frantic call from my program officer, the one who's now in charge of Zika at the NIH, and she suddenly went, Eva, oh my God, have you done cross-neutralization? And we're like, yeah, of course, cross-neutral. She's like, this is going to be terrible for like once we get the vaccines into Puerto Rico. I'm like, that's right. You know, so people, it's like you can understand it intellectually at some level, and then suddenly you start actually realizing what the implication of this is, and it's huge. Yeah, I mean, the vaccine studies that are going to be done in the United States, and they're pushing really, really hard on this and fast, will be fine. And then when you get them into, and remember, there's now suddenly a dengue vaccine that's been licensed, and it's going into a number of these countries. So now you have to contend with, you know, natural dengue infection with vaccine-induced dengue human immunity, and then Zika, and we don't have ways to tell them apart. It's very complex.
Okay, so we're getting near the end here because I wanted to give us a few minutes to talk. We'll have one more question back there, but first, one of the things that Marion alluded to in her talk was the speed with which the Puerto Rican blood supply was declared unsafe, and then we came out with the test. It was about a 6-week window there where we have a test under IND. And I know there are some people from Roche here. I can't find them in the crowd. They're great. So I just wanted to have a quick comment from you guys about how did you do it so quickly? I mean, this is really amazing, and this saved the Puerto Rican blood supply because they would not be able to collect blood now without a test. So what kind of resources went into it and decision making, and how did you do it? That's just one quick comment I want to get from you guys.
One quick answer. All of the resources went into it.
So what does that mean? I mean, for me, that's how 5 people last. What did you guys do?
Roche is a big company.
Yeah, so can you talk about that at all?
Well, so I'm from R&D, research. So it was the entire infectious diseases department, more or less, 30 people, about 40, 45 people in development. That's a lot of people.
Huge. Yeah. Okay. How long did it take from deciding to?
4 weeks for research and maybe 8 weeks total. Then regulatory gets involved, business.
That's incredible. Thanks.
Thank you.
We have one more in the back here.
Hello?
Last question.
I have a question about Wolbachia. So first about Zika, but also about dengue and other viruses more generally. I guess my question is that there's been a lot of laboratory experiments to look at Wolbachia in mosquitoes and how it affects virus transmission. And I know there's been some small field studies with mosquitoes and transmission, but my question is what's your opinion on how scalable this is, and if it actually is something that might actually, work on a countrywide scale? And I mean, if you can put aside all the regulatory kind of aspects of it, just is it actually better than just, the typical bed nets and dumping out all of the water sources and stuff?
So if you look at the Wolbachia-infected Aedes aegypti release data from O'Neill's group in Australia, where they did releases in Yorkneys Knob, which is a little town in northern Australia where they get dengue every year, they found that the Wolbachia-infected mosquitoes did not spread very well. And the magnitude of the flight range of these mosquitoes was something on the order of meters, like hundreds of meters. And they even found that if they would introduce Aegypti on one side of a road, they wouldn't spread to the other side. So I think initially the idea that you could introduce these mosquitoes and then have them spread themselves across a large area those ideas were sort of tempered by those pilot studies. So what they're doing now is releasing in lots and lots more places, and they're focusing on smaller scales, like in urban settings where transmission intensity is high. So if you could do many more targeted releases, I think the approach is still really viable. But it's not like, if you look at the story of Wolbachia and Drosophila in California, where it spread itself in a period of years, it's not going to do that in Egypti like we would have hoped.
I think it does matter. I mean, there's a couple strains that they've been working with, and so there's the MEL and the WML POP, et cetera. So it's the ones that have most effect against dengue are actually the ones that have the highest fitness costs. And so, but they're kind of, they now have some that have much better fitness and are able to drive into the population better. And so, and what they've been focusing on now is like as one very large study. Well, there's a number of countries where they're bringing it into urban centers, but in Yogyakarta in Indonesia, which is 400,000 people, there's a large study going on there. I mean, I worked a little bit with them on what's hard is like, how do you actually measure this? Because it really depends on not just the infestation of the mosquito, et cetera, but like the epidemiological outcomes. And that's very difficult because you can have differences in dengue, et cetera, et cetera. So with Zika, though, they've shown that, I mean, you do get the effect on Zika as well, and on chick as well. So I think that with like, if it is really 50 to 80% of the population that's going to get infected, there might actually be a good test run for, how this method can work.
And as an aside, releasing mosquitoes has social consequences too. I heard that there were rumors going around Brazil that Zika was actually brought in through mosquito releases because Wolbachia releases were going on in an area that was in Brazil, or sorry, Oxitec, so different drive, but same idea. So take that for what it is, but it's...
That mosquito is going to be FDA approved pretty soon for US.
Okay, well, we want to have a quick break before the lectures resume, so I have to cut off the questions. I'm sorry, but can we please thank the panelists? Awesome panel. Thank you so much.